being recorded and made available on the OTS website. And all webinar participants are muted to minimize the background noise. So if you have any questions, please put them in the chat box. And at the end of the presentation, we'll have a question and answer session where Andrea will do her best to answer your questions. Over to you then, Andrea.
Great. Thank you so much, Petra. So thank you all very much for joining on this sort of little bit of an introductory to design of microRNA therapeutics. And so just to get, I guess, everybody really a little bit of a foundation, I thought I would start the presentation with some background basically on the history or the trajectory of microRNAs, really from their discovery all the way to the bedside, and where we've gone so far since the discovery of the first microRNA by Dr. Victor Ambros' group in 1993 in this beautiful yet simple model system, C. elegans. And through a screen that Dr. Ambros did in his laboratory, they identified a lineage defect in one of their worms, and upon mapping the gene, determined that the gene mapped to a small RNA, and hence gave that Lin4. And at this point in time, it was thought that perhaps these microRNAs might be a worm-specific entity. And indeed, 7 years later, a second C. elegans microRNA was identified, given the name LET7 due to the fact that the mutation caused lethality in this simple model system. But what was very different at this point in time was that LET7 was determined to be or identified in the human genome. And so at this point in time, it was recognized that these microRNAs likely are not worm-specific, but indeed are conserved in other systems, including humans. The next 2 years really caused a breakthrough in the field when it was identified that there are many microRNAs in the human genome, and moreover that deletion or gain of function of some microRNAs, in this case 2 microRNAs co-expressed in a cluster miR15 and 16, were determined to be lost in chronic lymphocytic leukemic patients. And this wasn't just a correlation. These microRNAs were actually determined to contribute to chronic lymphocytic leukemia. So not only are these microRNAs identified and found in the human genome, but their misexpression certainly can be attributed and causal in various diseases, including cancer and others. So I want to focus the presentation really on 2 classes of microRNAs as we talked about the transition to generating microRNA therapeutics. And those classes are going to be reflected here in firstly, this microRNA miR122. So in my next slide, I'll talk a little bit about the background of miR122, but briefly here, it's a microRNA that's heavily expressed in the liver, and it coordinates to promote HCV infection. And that biology was determined in 2005, and in as short as 5 years here, an anti-miR or a small molecule that binds with perfect complementarity across a portion of miR122 was taken into clinical trial for the treatment of HCV. And again, we're going to focus a little bit more on some of the aspects of this particular anti-miR as we move through today's talk. In addition to inhibiting microRNAs, certainly there's diseases where microRNAs are lost, and then there's this question about whether or not you can use microRNAs for restoration therapeutics. And so the second microRNA that we'll spend a little bit of time on today is this microRNA miR34. So miR34 was discovered in 2007 as a microRNA that's actually lost in a variety of cancers, and part of that is due to the fact that it's downstream of one of the most mutated genes in human cancer, namely P53. And again, from its discovery in 2007, it took only 6 years to take miR34 into clinical trial in 2013. And so we'll look at, again, some of the necessities for advancing microRNAs into the clinic, what's needed, and then some of the things that we're still presently working on to advance these even further. So the first step in any sort of therapeutic approach, of course, is going to be to select the candidate. And I already gave you a little bit of background on these 2 candidates that we're going to be looking at today, miR34 and miR122, and they subclassify into 2 distinct categories. One are microRNAs that are going to be overexpressed in the the disease state, and therefore the goal of the therapy is going to be inhibit that microRNA. And you'll see over and over again through the talk that those are often referred to as anti-MIR therapies, where we're blocking the activity of these through sequestration. The other class are microRNAs that are reduced in disease state, as I mentioned for miR34, and these are ones that perhaps we want to restore, particularly if we can find out that overexpression of the microRNA or reintroducing this microRNA can help with whatever that disease, the disease is. There's a few other things that we like to consider when we're selecting a potential candidate that's not on this screen, but I do want to make mention of them. Of course, upfront knowing about their involvement in particular pathways is really important, because at some point we're going to want to go back and validate that these microRNAs or these anti-MIRs are actually functioning as intended. And so you need to know, of course, a little bit of information about the actual molecular biology that's going on. Other things to think about, and this is really important when we think about miR-122, this is an example of a microRNA that's limited in its expression in nearly every other tissue in the human body other than the liver. And so in the liver, it has a very high expression level. And so you can imagine that when you go in with a therapeutic and you're trying to sequester this microRNA, the potential for having off-target effects in other tissues is minimal as far as sequestering miR-122, since it's not present in those tissues. So it makes miR-122 sequestration in the liver a really great microRNA to think about based on its location. And then the other thing, as you're selecting a candidate, of course, this is not something that you have a lot of control over, but you do need to think about what tissue you're going to be targeting. And so with miR-122, the anti-MIR therapeutic is going to be delivered directly to the liver, and that's, of course, one of the easiest, albeit still challenging, but easier organs to deliver anti-MIR to. And that's not always the case, as we'll see for other therapeutics such as miR34, which will be delivered to tumor tissue throughout the body. Okay, so just a few slides on the rationale for why we would want to, say, inhibit a microRNA or perhaps restore the microRNA. And so again, using the models, in this case, for antagonizing miR-122. Most of you are probably very familiar with the microRNA targeting pathway, where most microRNAs bind to a target and inhibit that target gene expression. And that's quite different, actually, for miR-122. So it interacts with the 5' untranslated region of the hepatitis C viral RNA. And it has 2 binding sites for miR-122. So there's 2 seed regions shown here. And miR-122 binds in an argonaute-dependent manner. And as you can see on the lower picture, these are seed-dependent binding interactions. So we see that the microRNAs in these 2 binding sites are binding through the seed sequence, namely nucleotides roughly 2 through 8 of the microRNA. But binding, instead of inhibiting a protein coding gene expression, which is what we normally see from microRNA interaction with a target, in this case, it's causing stabilization of the RNA that encodes this virus. And therefore, when miR-122 is expressed at high levels, as is seen in the liver, this oligomeric complex of miR-122 complex to the HCV RNA protects this RNA from nucleolytic degradation, thereby enhancing viral production. And so the idea that Centaurus Pharmaceuticals had by generating an inhibitor, namely miroviricin, shown here on the right-hand side, is that through sequestering miR-122 away from the 5' untranslated region of this RNA, the RNA becomes basically destabilized, and therefore you lose the production of virus. And so we'll discuss a little bit later on about the design of miroviricin. In particular, as you can see here, it's not quite as long as the microRNA. It's only 15 nucleotides in length, and there's a handful of modifications on that RNA that help to stabilize it that we'll bring up in a few slides here. So that gives a little bit of background on the rationale for why we may want to antagonize an RNA. And you can imagine in your own context, perhaps an RNA that you'd be interested in antagonizing likewise. What about for situations where you want to restore a microRNA? And again, looking at miR34 as an example, So again, miR34, there's 3 family members. They're all downstream of this heavily mutated gene in human carcinogenesis, P53. And so in P53 mutated tumors, the expression of miR34 is greatly diminished. However, what was determined by a variety of groups with some very elegant science is that in tumors that have lost P53 signaling, tumors or cell lines, simply reintroducing miR34 into those cells or into that tumor tissue can restore some of that inhibitory effect on those oncogenic processes, namely inhibiting cell cycle progression, proliferation, and promoting apoptotic signaling events. And so it was reasoned that perhaps restoration of miR34 would be a valid therapeutic for individuals that have mutations in P53 signaling pathway. But this goes beyond just mutations in P53, and it's really useful to think deeply about the biology of the system that you're working on. For example, work out of George Kalin as a postdoc when he was working with Carlo Croce, where they were looking at fragile regions of the genome. And these are regions that are often either deleted, as you can see with these various Ds, or sometimes somewhat less frequently amplified in cancer. And so in these regions, these chromosomal regions where these deletions or amplifications occur, there were very few known protein-coding oncogenes or protein-coding tumor suppressor genes. And what George and Carlo found out was that within these regions are many microRNAs. And in particular, what was very interesting was there's a chromosomal region, 11q23, that is often deleted in both breast and lung cancer. And within that region were 2 isoforms of miR34. And so we reasoned that in addition to individuals that have mutations in P53 being responders to miR34 therapeutics, that perhaps individuals that have deletions of miR34, they may have wild type P53, but miR34 may be deleted. Or likewise, I didn't mention it, but miR34 is also epigenetically regulated and repressed due to hypermethylation, that these individuals would also benefit from miR34 restoration therapeutics. And so this just gives a little bit of a background on, again, a microRNA that perhaps would have a really striking phenotype with regard to tumorigenesis if we can restore it specifically in the diseased tissue. So we talked briefly about selecting a candidate, and then, the next step, of course, just doing some basic proof of concept in cell culture is really validating that this candidate is functioning as intended and would be a valid candidate as a therapeutic. So in the context of cancer, we would likely go to a cell culture model. That's not always useful depending on what the disease model may be. You may have to go to a simple model systems like zebrafish or C. elegans to validate some of this. But the idea would be that if you modify the level of that RNA via overexpressing it or antagonizing it through sequestration, that you need to observe some sort of phenotype. So somewhat very just basic biology here. And then validating that through perhaps Western blot, making sure that at the protein level, the particular targets that would be involved in disease are indeed altered. Certainly qRT-PCR, and maybe at a global level, RNA sequencing, which may also give you some insight into perhaps some off-target effects that may be occurring at a therapeutic level. Once you have some evidence on cell culture or in that simple model system, it's important then to move up to a mammalian model system. Of course, mouse models are usually the first one that we gravitate to, especially in the cancer world. And the easiest one in that context would be a xenograft model where you have the same cells or a certain cell type where you're overexpressing or knocking down that particular RNA. And this can be done in a variety of ways, perhaps transiently with transfection, but more importantly, and probably in a better way, would be virally, where you can actually overexpress a microRNA in its unprocessed state using a virus, and that can be stably integrated into your cell line. Or to sequester a microRNA, you can overexpress, again, through transgenic approach, a sequence with multiple binding sites for a microRNA, which we would refer to as a sponge, which would be involved in sequestering that microRNA away from its endogenous target genes. And then those cells can be implanted into the mouse model and the phenotype could be observed. Likewise, you can then move into genetically engineered mice if that's necessary, or perhaps for your disease model, genetically engineered mice aren't needed, but certainly an infected or a disease model may be important. And this is very important as we move forward, especially when we think about delivery, which we'll get to in a little bit here, with regard to being in an immune-competent model, so an immunocompetent model, making sure that we're taking into account the immune system on the effects of these RNAs as well as the delivery vehicle, so that we're dealing with kind of the whole package, if you will. And then at some point, many of the studies will advance into primate models prior to entering into human clinical trials. So a few things, and this is where really the bulk of the talk is going to go, is two really things that we need to consider as we advance microRNAs forward. And one is the stability. So a simple microRNA mimic or an integumer that you order from, say, QIAGEN or Exicon are great for cell culture experiments, but when we think about moving in vivo, we really have to be cognizant about the stability of these RNAs, especially in the context of the circulatory system, and making sure that they're stable enough to get into the cell, and considering the half-life and how often dosing needs to occur. So if we can think about some of these modifications for stability. And then the second thing we'll talk about in a little bit is delivery, which is equally, if not more important. So I'm going to go back for a minute here to the sequence of miR-Vericin that Centaurus Pharmaceuticals took forward for inhibiting miR-122. And I'll zoom in on a portion of that sequence to look at some of the modifications that are important as we move forward. So the first modification that is going to take place that's is one that's actually on the backbone. So this is a phosphorothioate or PS linkage. And what we're looking at here is one of the non-bridging oxygens that I'm circling here, of course, now is converted to a sulfur, but this was one of the non-bridging oxygens in an internucleotide linkage is replaced with sulfur. And so this change creates modest reduction in binding affinity between the microRNA and the target, but with the gain of two really major benefits. This molecule with these PS linkages is substantially more stable towards hydrolysis by ribonucleases compared to unmodified RNAs. And another interesting point to bring up is that PS modified oligos have higher affinity to plasma proteins. And what this does is it reduces their clearance and excretion. So if these molecules are delivered in the absence of a delivery vehicle, i.e. naked or free in circulation, they tend to get cleared rapidly due to their small size. So the fact that the PS modified oligos have an affinity for a plasma protein, that helps to reduce their clearance, and therefore you get improved pharmacokinetics. And so kind of capitalizing on some of the work that has been done with siRNAs, it's well recognized that to improve the stability of the siRNAs that are out there, usually one PS linkage, at least one PS linkage is introduced at the 3' terminus of both the sense and antisense strands. And so a second modification that we see here for increasing stability specifically for antagomeres is what's referred to as a locked nucleic acid, and that's shown here as well. And what we see is that at the 2' position, we have a bridge or a locked nucleotide, if you will, to the 4' carbon through this methylene bond. And that prevents these molecules from being degraded as well, generating resistance. So one of the things about locked nucleic acids that's interesting is they possess very high thermal stability when they're hybridized with their cognate target molecule. So you get increased stability between the microRNA and an antimer, for example. And so this allows us, and this allowed Centaurus Pharmaceuticals to generate a much smaller molecule. So we're not talking about a 22 nucleotide antimer here, but instead we're looking at a 15 nucleotide molecule due to the enhanced thermostability. And this is being capitalized even further in the field where there are anti-miRs that are roughly 8 nucleotides in length. And the benefit to those molecules is that you can generate a single locked nucleic acid anti-miR that targets an entire family of microRNAs by just really targeting the seed sequence. And because of that high thermal stability, a single anti-miR can target the entire family. So that's really a great advantage here. LNAs are great for increasing the stability of anti-miRs. Unfortunately, they can't be used for increasing the stability of microRNAs, and that's due to the fact that they don't allow for the promoting cleavage, which is essential for microRNAs, as we know. So some of the stability modifications that can be done for a microRNA are certainly a little less than what can be done for an anti-miR. One of them that is in common is this PS linkage. So again, the PS linkage is very commonly used in microRNAs to, again, enhance the stability, preventing nucleolytic cleavage. And just to kind of remind you, with regard to an RNA molecule, there's that free 2' hydroxyl group, which is subject to hydrolysis. And so a few additional modifications that occur in microRNAs to enhance stability are modifications to that 2' region. And so you'll see 2' O-methyl groups, 2' floral groups. These are probably two of the most common, and 2' O-MEOs here as well that are used for microRNAs. And one study that I want to bring out is a study published a few years ago, just to highlight the fact that in this study they had a fully modified version of miR34A. So each of these nucleotide, the colors correspond to the colors shown here for these different modifications. The S's in here dictate whether or not there's a PS linkage in there. And they compared this fully modified miR34 to a standard double-stranded microRNA mimic that you might get from one of the standard biotech companies. And what was observed here, what was really exciting, was that the fully modified miR34 was able to downregulate its target nearly as well as the unmodified duplex. And so although we don't have any information in this particular study regarding stability, the idea that this fully modified microRNA is still able to target its intended target gene and downregulate the protein production is a step forward, and argues that these microRNAs certainly can be fully modified, similar to what we're seeing for some of these siRNAs out of Dr. Anastasia Kavorov's lab and at UMass and Alnylam, as well as some other researchers. So this is really helping to advance the field forward. So this is all I'm going to mention about stability right now. I think this is somewhat of an open-ended question in the microRNA field that we're still working towards with regard to increasing some of these modifications so that we can further stabilize these RNAs to not only reduce dosing, but save dollars, if you will, as we move into human trials due to the reduced dosing. So this is likely one of the biggest bottlenecks in the field, this next area that I'm going to talk about, and that's identifying a suitable delivery vehicle. You can have the best fully modified microRNA with great activity in a cell, but if you can't get it to your intended tissue, then you really have nowhere to go as far as a therapeutic approach is concerned. And so I'm going to subclassify these delivery vehicles into two classes. One's going to be essentially a class that we refer to as protected or encapsulated, and the other class will be the unprotected class. So the encapsulated class is probably historically the most commonly used. That includes lipid-based molecules, polymeric molecules, and more recently exosomes. And for some of the lipid-based ones, this is the class that was taken into clinical trial with miR34 in the context of a SMARTicle. And many of these encapsulated lipid-based particles have a anionic nature in basically in circulation, but when they encounter an acidic pH, they convert to a cationic or positively charged molecule. And so this allows these particles to be taken up specifically at low pH, and that's a very common occurrence in the context of the tumor microenvironment. And so you get somewhat specific uptake, albeit still lacking in being able to target micrometastases that perhaps have not set up this acidic microenvironment. There's other things that can also be done to these lipids as well as polymeric molecules. Certainly lipid modifications have been used to deliver RNAs to osteoblasts, and peptides have been used for delivery to endothelial cells, as well as a variety of other linkages and decorations that can be added to these molecules. So I'm going to move forward to what we refer to as the unprotected group, and these will be subclassified into a few different categories. So the unprotected group, what we're referring to here is that these RNAs are not encapsulated. So they're completely free, if you will, in circulation, and there's kind of two classes of those. There's the ones that we refer to as unconjugated, meaning that it's just the modified RNA that's going to be in circulation, and uptake is usually going to be via a process of gymnosis, and that's really uptake in the absence of any carrier or conjugate. And this is usually only going to be the case for anti-miRs because they can be fully modified, stabilized with these locked nucleic acids to protect them, as well as remove the negative charge so that they can freely traverse the plasma membrane. Some of the more common ones that get towards more targeted approaches entail targeting these to some sort of ligand. And so there's ones that are considered non-targeting, for example, cholesterol, and that's going to be used for delivery to the liver. So in some ways it may be slightly targeted, but not as specific as some of the other targeting moieties that we'll discuss here in a few minutes. And one of the interesting ones that I have on here too is a cell-penetrating peptide referred to as a FLIP anti-miR. And this one is a pH-dependent peptide, in that this peptide here in low pH, again, something that occurs in the tumor microenvironment at a pH less than 7, the C terminus is going to insert across the lipid bilayer after it forms this inducible sort of transmembrane alpha helix under these acidic conditions. And when the C terminus does that, it brings with it this conjugated anti-miR, in this case anti-155, that will enter the intracellular space, and through a disulfide linkage, you get liberation of that anti-miR to go in and then target its complementary microRNA. And so these are some of the what we refer to essentially as the non-targeted class. And then there's ones that are more of the targeted class, which are highly specific in targeting. And there's a couple criteria that are really important as we move forward with specific targeting. You can imagine many benefits to this. For example, you can dramatically reduce dosing. And for example, this really depends on the affinity of the interaction. And so if you have a drug conjugate, let's say, that has nanomolar affinity to occupy all the targeted receptors, in order to get a similar response with a conjugate with micromolar affinity, you're going to have a 1,000-fold higher concentration of drug. And so the specific conjugates that I'm going to mention today as far as delivery are all in the nanomolar range, and so very high affinity interactions. A couple other things that are important, recycling of the receptor certainly helps so that you can get repeated dosing. So if that receptor gets intracellularized and then makes its way back out to the plasma membrane, it can bind to another conjugated molecule. Abundance of the receptor, really important here. Of course, accessibility goes without saying, but as far as abundance, when you're delivering a ligand conjugate, you only have one molecule delivered per ligand. That's very different than if we're delivering a nanoparticle like a lipid or a polymer, where you can have 1,000 copies of that RNA being delivered per nanoparticle. In this case, we have one molecule per ligand. So the receptor needs to be at high enough concentration on the target cell And of course, that recycling helps too in such a way that you get enough therapeutic quantity into the cell with that single ligand conjugate. So a few examples of specific targeting that some of you may be familiar with. Of course, there's classes of glycoconjugates. The most historic conjugate probably that's out there right now is GalNAc. That's made its way into clinical trials with pharmaceuticals for delivery of some of their siRNAs, as well as Regulus. And shown here is a cartoon of GalNAc conjugated to an anti-miR-122, again for sequestering 122 in the liver. And GalNAc will bind to a receptor specifically in liver cells. So again, we're targeting directly to the liver with this GalNAc conjugate. Some of the other types that are interesting are aptamers, and there's actually a webinar I saw that's coming up on aptamer delivery. So if that's of interest to anybody, I encourage you to look at that. Aptamers have been designed, for example, to prostate-specific membrane antigen. So high-affinity aptamers for that have been used to deliver tumor-suppressive microRNAs, such as miR-15 and miR-16, to cancer cells, as well as various anti-miRs. And then aptamers to AXL have also been developed, very high-affinity aptamers. So AXL is a receptor tyrosine kinase that's overexpressed on cancer cells, and has been the aptamer conjugated, the AXL aptamer has been conjugated to tumor suppressive LET7G, which has been used to deliver this tumor suppressive microRNA to cells overexpressing the AXL receptor. And then small molecule conjugates, really important class here. Some of the advantage of these small molecule conjugates, typically they're very easy to synthesize. Following delivery, they can be broadly distributed. They're small enough that they can penetrate the dense architecture of the tissue or the tumor microenvironment. You have somewhat greater selectivity because you kind of have this lock and key mechanism between the ligand and the receptor. And they certainly tend to be a lot less toxic than some of the nanoparticles, which are composed of multiple components. In this case, some of these small molecule components entail really a single molecule, and I'm going to tell you a little bit of a story about one of those that we worked on with regard to folate, which is a molecule that's essential in the diet. So as far as toxicity is concerned, our hypothesis initially moving forward was that we would see very limited toxicity with this. And so the idea behind a ligand conjugated delivery vehicle in this case is based on the fact that in certain cell types, if you find a receptor that's overexpressed in a particular disease that's overexpressed or perhaps inconsequential for targeting in non-disease cells, then that can be used to deliver something that is conjugated to a high-affinity ligand, in this case, folate. And so folate binds with, as I mentioned, nanomolar affinity to the folate receptor. And in a variety of cancer cells, the folate receptor is expressed at levels of approximately about 10 to the 6 copies per cell. So we certainly have achieved the number of receptors that would be necessary to get a therapeutic dose in to the cell. And so we conjugated a warhead onto this, in this case, miR-34, and decided to start with 2 different chemistries that I'm showing you here for that. And so on the left-hand side in blue is again, the very small molecule of folate, very easy to synthesize, and this is synthesized with a DVCO, so that through click chemistry, we can conjugate onto that the passenger strand for the microRNA of interest, in this case, miR-34. And then, and you can see here with these M's that some of these nucleotides are modified on the passenger strand, roughly every other nucleotide. And then we anneal the mature strand directly to that, and the mature strand only has 2 modifications on the 3' end, so there's 2 methylated residues there. And so the 2 chemistries that we move forward with here were one with a disulfide bridge that we hypothesized would be reduced in the early endosome, and that may be essential or necessary to liberate away the folate from the rest of the molecule so that we get efficient loading into Argonaut. And the second molecule, of course, does not have that disulfide bridge. So in this case, there would be no reduction and no liberation of the microRNA. Moving these forward, I'm going to show you a little bit of data from this study, as well as some things that should be taken into account as these molecules move forward, any molecule for that matter, any microRNA moves forward clinically. And this has to do with some of the pharmacokinetics and the pharmacodynamics. So of course, the first thing that needs to be considered is the therapy needs to get to the intended tissue, right? And that's really the highlight of the delivery vehicle is, can the delivery vehicle get it to where you intend? And I'm showing you some data here on the right-hand side that certainly shows that folate conjugates, this isn't the microRNA, this is a folate conjugated to a near-infrared dye, but nonetheless, this folate conjugate very nicely localizes to the tumor. So if we look at the H&E stain, we get very nice overlap with the NIR signal. And importantly, we're getting very little signal coming from any of the normal tissue, kind of shown in this region number 2 here. And we've done this as well in animals that don't have tumor, and we see absolutely no uptake whatsoever in the lung. So making sure you get to the intended tissue. Although that's not the only challenge, getting to the tissue doesn't necessarily state that you've actually penetrated the plasma membrane, which is inherently difficult for these negatively charged RNAs. So that's kind of the second point here is, can you get it into the cell through the plasma membrane? And in doing so, can you get it to the correct compartment? If you need that RNA to be in the nucleus, is it in the nucleus, is it in the cytoplasm, or is it sequestered somewhere else without bioactive activity? So fortunately in the microRNA field, we have a really elegant way to monitor this through various reporters. So in this case, using a Ranilla porter with a microRNA binding site downstream. And so if the microRNA binds to that site, we'll get reduction in Ranilla signal. And so cells were generated to stably express this, and the data I'm showing below here is from xenograft models, where these animals were injected with a single dose of either of these microRNAs. So one is the disulfide bond and the other one without the disulfide bond. And a single injection, just the one without the disulfide bond, resulted in this really nice reduction in Ranilla activity. And this was systemically administered to animals with this xenograph, and then of course the Ranilla activity was monitored. So a few things to point out here. So we know that we got it, of course, into the cell of interest because our cell had the reporter. We know it was into the correct cellular compartment because our Ranilla reporter was in the cytoplasm and responded beautifully to that. One of the things that is a concern here is this concern about circulation half-life, and we certainly have not achieved that because we get this rebound in activity after about 48 hours. So we know that this single dose was not stable enough to maintain itself in circulation or perhaps also within the cells. The other thing, just because I see some questions coming up that I just want to mention, is that I don't have time to tell you this, but the disulfide version with the cleavable linker did not work in vivo. It worked very well in cells in culture, again, highlighting the fact that you really need to perform these studies in cell culture, but not always does cell culture mirror what's going to happen in vivo. And so the reason for that is that disulfide was being reduced prematurely. So basically the bridge was cleaved prior to the microRNA getting to the cell and gaining entry to the cell. So the microRNA never made it there. So as far as the circulation half-life is concerned, some of the things that are being accomplished in the microRNA field right now are, as I mentioned before, inclusion of these PS linkages, which perhaps will reduce renal excretion and help these very small molecules that get excreted rapidly to maintain themselves in circulation as well as stabilize themselves so that we get a more consistent response as what's been observed for the siRNA field. Okay, so what about the distribution of the microRNA? This is another thing that's really important and gave us some insight on this disulfide-linked RNA. So I'm showing you here some infrared images on the top right of where the conjugated RNA was distributed within these animals. And for all of them, a very nice near-infrared expression in the tumor, whether the disulfide linkage was there or not, because the NIR was actually conjugated to folate, not to the microRNA. We also see some signal coming from the kidney, not surprising since the kidney does express levels of the folate receptor as it scavenges folate from the diet. Importantly, we didn't see a lot of uptake in the liver, and this was great for us because there's a lot of agents that will deliver to the liver, but very few extrahepatic delivery agents, so we were excited by exclusion from the liver. So we saw distribution there. Another way to look at distribution was to actually harvest the tumors and do QRT-PCR on those. And when we did that, we only saw miR34 expression increased when we had the unconjugated or the molecule without the disulfide. And so that gave us some evidence that this disulfide version, the miR34, was never actually making its way to the tumor tissue. And so after looking at sort of the distribution, then of course we move on to more of the pharmacodynamics, which entails really the targeting and some of the mechanism. And so what we're looking at here is in red, and I apologize, I'm not sure if I can get rid of that. There we go. So in red here are cells that were treated with our folate conjugate with a near-infrared dye attached. And we did flow cytometry analysis, and you can see that there's that shift up here in fluorescent signal to the right, arguing that the cells were taking this up. But the question was, mechanistically, when we think about pharmacodynamics, was this happening through gymnosis, or was this happening through the folate receptor? And so through competition studies where we're competing with folate glucosamine, you can nicely see that we get a shift that slowly moves its way back to the control setting, arguing that we were mechanistically going up through the folate receptor. Moving further with some of the pharmacodynamics and looking at the targets of these microRNAs, so harvesting tumor tissue out and asking, were we actually downregulating miR34 target with the BCL2? We saw a nice reduction in QRT for that, as well as MYC. MET was not statistically significant. But we did see downregulation at the transcript level. Other things that you can imagine doing here would be looking at immunofluorescence, immunohistochemistry, or perhaps Western blotting if you have enough tissue to do so. You can also perform RNA sequencing, which is also very useful to look at whether or not you're altering the targets and perhaps, again, if there's any sort of off-target effects that might be useful to look at. And so just to kind of follow up here and finalize on mechanism and really activity more so. So delivery of this folate conjugate, in this case, we're only looking at the molecule without the disulfide. The other one didn't have activity in vivo. We see a really nice response in the ability of this molecule to repress tumorigenesis in this lung cancer mouse model. So on the top right are images of thoracic cavities taken by MRI for animals administered the negative control throughout the course of the treatment at day 8 and day 29, and you can really appreciate the change in tumor burden in these animals, where the animals treated systemically with this miR34, you can see this nice reduction, and all that data is quantified down here below. And so it was pretty exciting, although we also have to think about toxicity here, and I can at least state that for the folate conjugates, we still have not been able to get these into a dose that's high enough to observe any sort of toxicity. But it's certainly important to continue trying to do that through multiple dosing studies, which we have yet to do for these, really important. We've only done single dose MTD. Also, immunogenicity. So again, doing these studies in an immunocompetent model system, looking at some cytokine complement, other things that could perhaps lead to any sort of toxic events, doing these studies also in peripheral blood mononuclear cell culture, really to hone in on any of these perhaps toxic side effects that may be occurring. So I want to kind of stay on this delivery for a little bit, because I don't think we've tackled the holy grail here, and I think as a webinar that's informative, it's really important to talk about some of the other things that we need to consider as this field moves forward. And I'm going to talk about two of them that are specific to targeted delivery, and then I'll talk in a few minutes about sort of this idea of dynamic cellular events that could alter some of the targeting, and we need to be cognizant of that. So firstly, when we think about targeted delivery, heterogeneity is really critical here. So we have a really great specific ligand conjugate that's going to deliver to cells that express high levels of a receptor. But for example, the tumor tissue or many disease states, the disease tissue has some heterogeneity with regard to the receptor expression. And so that's a really big concern, whether or not you're going to be selecting for cells that express lower levels of the receptor. And so right now, one of the things that we're interested in evaluating is whether or not we have a bystander effect, where cells that have high levels of expression of the receptor can co-distribute the microRNA to the surrounding cells, the few surrounding cells that have lower levels of the receptor. Nonetheless, this is really an important concept that we need to think about as we move forward with targeted delivery agents and perhaps selecting for resistant cells. The other one has to do with uptake. So the majority of these targeted delivery agents are taken up through this process of receptor-mediated endocytosis. And so that's another area that we're working on, and I think many labs are working on. And so I'll take you through a few of the slides on that as well with regard to our folate conjugates. So as I mentioned, the folate conjugates are going to bind to these high-affinity receptors on the plasma membrane, and they're going to be internalized through a process of receptor-mediated endocytosis. And the inside of the endosome mirrors the extracellular environment in that it has high amounts of sodium ions, while the cytosol has high amounts of potassium ions. And usually as that early endosome makes its way through the endocytic pathway, it exchanges sodium for potassium, generating an electrochemical equilibrium inside of the endosome. And so we wanted to capitalize on that, taking advantage of that distribution inside of the endosome with these solutes. And I'm just showing you here a confocal image to show you that indeed, if we fluorescently conjugate folate to a Cy5 fluorophore, we monitor where it's going in the cell, we certainly see a lot of these punctae that are reminiscent of these endosomes. And then what we also see is this very strong signal from the periphery, and you can see that here when we quantify this, looking at the pixel intensity. And what that tells us is that a lot of our microRNA is sequestered inside of endosomes, and a lot of it is being recycled back out. And of course, that's what we don't want to do. We need to get it into the cytoplasm. We know we get some there, but we think we can reduce dosing much more if we can get a lot more of this out of these endosomes. And so how are we doing that? There's a variety of mechanisms that are being tackled for this. The one that we're working on right now is an intramolecular conjugation of a small molecule ionophore called nidriacin to our microRNA folate conjugate. And what nidriacin does is when it enters into the endosome, it inserts itself into the endocytic membrane, and it allows for the internalization of potassium ions, and potassium ions are osmotically charged, so they're going to bring water into the endosome. But instead of exchanging it for sodium ions, which are also osmotically charged, there's an exchange for a proton, and protons are not osmotically charged, so they're not going to take water back out, so we're bringing water in without taking water out, therefore causing the endosome to swell and redistributing the microRNA to the cytoplasm. In this confocal image, you can appreciate that there is redistribution. And we recently published on this, and the rest of the data are included in that manuscript that highlights that we're getting a lot more into the cytoplasm. It's bioactive. It works much better than the first generation folate conjugates. And we see a lot more engagement into Argonaut as well, and it's an Argonaut-dependent process. So that's one of the things that we certainly need to continue to work on in the field if we're thinking about these targeted delivery vehicles. And then finally, the last thing to consider is that, and this is true for any microRNA or any integument that we're considering, is that the targeting is going to be extremely dependent on these really dynamic events that are going on in the cell. And so for starters, you have to consider all the different microRNA or all the different messenger RNAs or RNAs in the cell that have the ability to sequester your microRNA. You may be perhaps interested in just targeting maybe this blue open reading frame coding gene. But if these other transcripts are expressed at a high level, they're going to be sequestering your microRNA away from the intended target. And this isn't just true for messenger RNAs, but this is also true for pseudogenes or other competitive endogenous RNAs, like long non-coding RNAs or more recently identified circular RNAs that could be really pulling that RNA away from the intended targets. And this is one of the things that the cell may absolutely be using to generate resistance to some of these microRNA therapeutics. The other things, I kind of have here listed as stability of microRNA binding sites. So on the top of this diagram here highlights the fact that many genes utilize different polyadenylation signals. So if you have a gene that uses the distal polyadenylation signal, which is shown over here, you conserve all your microRNA binding sites. And therefore, if you're putting in a microRNA, a therapeutic RNA that you believe should target this gene, it'll likely have an effect. But there's many genes, especially oncogenes, that have used the proximal polyadenylation signal. And in doing so, what you can see up here is they've basically depleted the microRNA binding sites. You may be putting in a microRNA that you intend to target MYC or one of these other oncogenes, but the targeting may not be as robust due to the fact that the cell or that may be using an alternate polyadenylation signal for that transcript. And then the other kind of stability issue has to do with the fact that microRNA binding sites can be mutated, and those mutations can perhaps induce a microRNA to bind where it shouldn't, or prevent a microRNA from binding. And one of the probably key studies that found a mutation in a microRNA binding site is for LET7 binding to KRAS. That has been pretty well documented. So those are really some of the things that I think the field needs to consider moving forward, and some of the ideas with regard to microRNA therapeutics. I just wanted to bring up my last slide regarding some of the people that help with this work, and I would be more than happy to continue to address some questions that you have. Okay.
Well, thank you very much, Andrea. That was a very enlightening talk. There are a few questions already, but I think you've answered some during your talk.
Okay. Yeah, so I'll go ahead and I'll start reading through some of these and continue to answer them. So it looks like Flip has a question about the melting temperature for miraviricin. I honestly don't know that offhand, but I can tell you miraviricin had, I believe it was 8 LNAs out of 15 in there. Inclusion of the LNAs will enhance the melting temperature, sometimes greater than 75 to 80 degrees. And so that thermostability really is increased. This is something that, well, I'm going to say somewhat of a challenge when we think about some of these modifications to microRNAs and thinking about a duplex. Now, LNAs wouldn't be used in a microRNA, but some of these other modifications also increase the thermostability. So these are other things that we need to be cognizant about, but unfortunately I don't know the exact melting temperature of miraviricin. And as far as the sequence-related off-target effects for miraviricin, again, not something that I know that much about down to the specifics, but based on the fact that miR-122 is only highly expressed in the liver, those off-target effects certainly would be constrained to the liver. I know that the safety profile of this antagomer is very good, and therefore likely very few off-target effects, and if they are there, they're not likely ones that are causing any sort of toxicity. And again, more questions on the miraviricin. Which method was used for checking promiscuity of miraviricin? I would have to look a little bit more into that literature, and I apologize for not knowing a little bit more on the exact details of that. So I'm sorry for not having all the answers for that, Flip. Hassan, does folate have off-target effects as well? Since we're relying on overexpression but not exclusive expression. So there's two parts to this question. So does folate have off-target effects? So as far as folate is concerned, I mentioned that folate is essential in the diet. So all cells require folate, and therefore folate receptors or lower affinity means to bring folate into the cell are definitely included on cells. So there's certainly possibility that the folate conjugates can be taken up by cells that have folate receptor expression, albeit the receptor is going to be at a much lower level, likely inconsequential for targeting, especially if we're delivering through the bloodstream, because these cells have polarity. A lot of times the receptor for folate is not going to be found on the side of the cell that's in context with the bloodstream. Now, as far as off-target effects with regard to the microRNA, we have not yet done any deep analysis as far as RNA sequencing is concerned. All I can tell you is that in our in vivo system, when we dose at the highest dose that we can get this in, which is 130-fold over the dose where we see a very nice effect, we see no toxicity. The argument, I think, one of the things I didn't mention is using microRNAs as therapeutics is that because they are endogenous to the cells, most of the other cells in the body are going to have some level of expression, and even getting a little bit in likely isn't going to have deleterious consequences. If we have off-target effects in the cell that we're entering in the context of cancer, if it's going to affect the growth of the cell, that's something that's actually okay for us. Now, if we're thinking about a neurodegenerative disease or something of that context, I think we'd have to be more cognizant about some of those off-target effects. Does it have to be a cleavable bridge? I already addressed that, so hopefully that has been cleared out. In fact, the cleavable bridge was not useful in our case. Does it have to be double-stranded? So we haven't tried to use single-stranded. I think using the double-stranded nature helps to stabilize and protect the RNA. The one report that I did mention that looked at that fully modified miR34 did so as a single-stranded molecule. However, this was done via transfection. So again, stability was not addressed. I think this is somewhat of an open-ended question and will depend on how many modifications we really can make to these microRNAs. Certainly the double-stranded nature helps to protect them. The one with the SS bond did not work in vivo. And so why is tumor burden increasing in time? So I don't to argue that these microRNAs are going to be the magic bullet. What I think we're doing here is stabilizing the tumor formation. I will make mention that tumor burden is absolutely increasing in this mouse model. This is the most aggressive mouse model of lung cancer that exists, and it has yet to be cured by any FDA-approved agent that's already on the market. So the fact that we have somewhat stabilized the disease, at least initially, and have increased the latency of tumor progression in this model highlights the effect that these microRNAs will have. And I think we need to think more closely about how these microRNAs can be used as therapeutics, for example, maybe in combination with other entities, which is some work that we're actually following up with in my lab. So a question here about niduricin being an inflammasome inducer. Are we concerned about generating an inflammatory response? Absolutely. I didn't show you any of the toxicity profiling from our niduricin conjugates, but again, very safe. And the reason for that, we think, is because they're conjugated. And so we don't have free niduricin in circulation. And if we did, we are 2 orders of magnitude under the toxicity of free niduricin in circulation. So it's certainly something that is a concern and one that we have looked into and are continuing to look into. From Mark, what is your opinion about the chirality of PS compounds? I don't have one at this point. I'm sorry for that. We have not started working very diligently with PS in our molecules. It's something that we're going to be working on in the future, and so that's something that we need to consider as we move forward. And let's see, just want to make sure. LNA is not tolerated in microRNA therapeutics. That's correct. So the LNA doesn't allow for cleavage, and of course, a microRNA mimic or a microRNA needs to engage into the RNA-induced silencing complex, bind its target, and that target needs to either be cleaved or degraded or deadenylated, and the LNAs do not allow for that to occur. So LNAs are primarily used in anti-miR therapeutics. The PS is probably going to be the best backbone modification for microRNAs, and then, of course, some of these 2' modifications as well. If there are not any more questions, I imagine we're close to time here. Petra?
Yes, pretty much, but if we run over a little bit, I'll just have some closing comments then. Thank you again for doing this. This was very informative for me as well.
Thank you.
So the next webinar, if you're interested, will be in July. The exact date is yet to be confirmed, and it will be Donald Burke Aguero and David Porciani from the University of Missouri on target delivery using Aptamer. So we should fit in quite well with this one. Thank you very much again, Andrea. Very much appreciated that you're doing this for OTS. And
thank you all for joining.
Yeah. Bye, everyone.